It's very short, very simple sequences of DNA, like G, T, T, A, C, but just repeated over and over and over again at the same location. Here's essentially a picture of it. If you look at some spots in the human genome, here you can see GTTAC, 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 blah, 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 blah. In some cases, for thousands and thousands and thousands of repeats of this same phrase, so to speak, over and over again in a row. And so notice the distinction between this and the large segment duplications. The large segments were around 5,000 nucleotides, 5,000 different nucleotides, not a short repeating sequence. And that large segment duplication, they were not found one right after the other, right after the other at the same spot. They were on different spots on the same chromosome or on different chromosomes entirely. But these are where you find a very, very short sequence, but it's in the same spot, just repeated over and over and over again. And they're not 100% sure what these tandem repeat DNAs are for, but they have a pretty good idea. And to show you what, or to explain what this idea is, here's a chromosome. And remember there's this region, not necessarily in the middle of the chromosome, but usually fairly near the middle, called the centromere. And that's where the two sister chromatids are held together. They find a lot of this tandem repeat DNA around this region of the chromosome, around the centromere. And they also find a lot of this tandem repeat DNA at the very tips of the chromatids. And these are called the telomeres. Telo means the end. So in other words, here and here and here and here, you also find some of this tandem repeat DNA. And so what they think is going on here is this, that there are some special proteins that have to bind to the chromosome in the centromere region, and there's also some special proteins that need to bind to the chromosome at the telomeres. And so maybe these special proteins key in on this particular sequence to know where they're supposed to bind to the chromosome. As a matter of fact, it may go slightly beyond that. Some people have postulated that this same sequence repeated over and over might allow for some weird kind of base pairing that's not present in regular DNA, where you get lots more variation in the sequence. And so maybe it's not a normal double helix, maybe it's some variation on the double helix that these proteins bind to. Now, this is kind of a review question. Who remembers anything about the centromere? Like, other than the fact that it has proteins that hold the chromatids together, what else attaches to the centromere? Does anybody recall? The spindle fibers, yeah, very good. When the cell is undergoing mitosis, it has to maneuver the chromosomes around, like in metaphase, they get maneuvered to the middle, the equator of the cell, and in anaphase, the spindle fibers pull the two chromatids apart. Those chromatids attach essentially to the centromere region. Well, there's actually a region called the kinetochore, Gesundheit, which is the kinetochore is a protein complex on each chromatid, but right at the centromere region. And the kinetochore essentially is the socket where the spindle fibers plug into. And so anyway, so they think that maybe that sequence is what the kinetochore proteins look for, so they know where to bind onto the centromere region. And there's an interesting story about the telomeres, the ends of the chromosomes also. Imagine this is part of the chromosome. This is the end of the chromosome. Remember when chromosomes duplicate, the two strands move apart like this, and you get something called a replication fork, like that. And then, remember, there are these enzymes called DNA polymerase, or actually DNA polymerase 3, that has to make the complementary strand, so you get duplication. So here's the complementary strand that DNA polymerase makes there. And remember, that one moves in that direction. And here's the complementary strand that the other DNA polymerase makes in that direction. Well, what's tricky about the telomeres is this. Remember, DNA polymerase 3 needs a primer to begin transcription, not transcription, but to begin duplication of the chromosome. And that's fine. A primer can be made right there to have that segment go like that. But since this is the end of the chromosome, we can't have a primer just floating out there in space. And so this DNA polymerase right here on this strand, I guess that's the leading strand, has a problem. There's just nowhere to prime it. And so there's some special enzymes called telomerases that fix this problem by essentially just adding some extra DNA. Every time the cell duplicates its chromosomes, these telomerase enzymes fix this problem so you don't get a shortening of the telomeres with every cell division. Anyway, the point I'm making here is there are some special proteins like telomerase that need to operate at the tips of the chromosomes because of this problem of not being able to prime the replication at those ends. And so again, they think that these proteins key in on these simple sequences. Alrighty, and in addition to calling this stuff tandem repeat DNA and satellite DNA, it's also sometimes called constitutive heterochromatin. Now, constitutive means always. Like someone who's always late, you can say, oh, that guy's constitutively late. Who remembers what heterochromatin is? That's a word we talked about a day or two ago. Yeah, well, that's what this is. Yeah, it is the DNA on the chromosomes that's very tightly bound to the histones. Yeah. So usually, remember, the centromeres are I should say the DNA in the chromosomes is usually not very tightly bound to the histones. All throughout interphase, it becomes euchromatin, which means that it mostly unwinds from the histones. But some parts of the DNA don't do that. They stay tightly bound to the histones all throughout the mitosis cycle, all throughout all the stages of cell division. And these tandem repeat DNAs are usually like that. They usually stay very tightly bound to the histones. So anyways, it's kind of interesting stuff. And so, but it probably has some function in having proteins bind to it. Okay, but the biggest chunk by far of our genome is a stuff called transposable element DNA, which is sometimes called lines. The line part is an acronym. It stands for long interspersed nuclear, because they're in the nucleus, elements. And when they say elements, they essentially mean sequences in the DNA. So long sequences in the DNA that are interspersed throughout the nucleus, throughout the DNA in the nucleus. And here's what they are. This is kind of mind-boggling. They are jumping genes, not in the sense they allow you to jump in the air, but they are segments of DNA that actually move about in your genome, as strange as that seems. Now, in some cases, the movement is where they actually cut themselves out of one chromosome they're in and physically transport themselves to a new location. And in other instances, the piece of DNA makes a copy of itself, and then the copy is what goes to another location in the genome and inserts itself. I think of the distinction on when I'm typing on my computer. If I do Command, if I highlight a word and do Command X, then I physically cut out that word and I can paste it somewhere else. But if I do Command C, I copy that word and I can still paste it to new locations, but the original stays where it is. So this seems totally bizarre, and it wasn't discovered until, well, fairly recently, the 1940s. This woman here was the discoverer. Her name was Barbara McClintock, and she was studying corn, the genetics of corn. And when you look at an ear of corn, each little kernel you see comes from from its own separate fertilization event. So this kernel came from one sperm and one egg fertilizing each other, and this kernel came from a different egg and sperm fertilizing each other, and this one and this one and this one, like that. And so it's essentially a Mendelian crossing event, and each kernel is one of the offspring. So she was studying the inheritance of some of the pigment patterns in the corn kernels, and she saw some strange results that didn't really fit in with Mendelian genetics. And she was trying to explain these to herself, and the only thing that really fit the explanation was that parts of the genomes were jumping around within the genome of some of these cells. And nobody believed her at first. It just seemed such a bizarre idea, nobody was willing to accept it. And unfortunately, there's probably a bit of sexism also. They probably said, oh, she's just a woman, what does she know about science, blah, blah, blah. Well, it turned out she was exactly correct, and not just with corn. Once they started looking for these things, they found them in every organism. The bacteria have them, plants have them, animals have them, fungi, you name it. Everybody's got these things. And she eventually won a Nobel Prize for it. It took around 40 years for her to be recognized and awarded, but she did become a Nobel laureate. And since that time, they've done a lot of investigations on these transposable elements. And they found they actually come in two major families. There's the big group, and one type are called transposons. And the other type are called retrotransposons, which does not mean they dress up like they're from the 50s with snake back hair and stuff like that. They essentially have reverse transcriptase is where that name comes from. Like so. Okay, so that's not it. Here we go. The actual official distinction between the two types of transposable elements is this. Transposons, when they jump around in your genome, do so by means of a DNA intermediate. It's a piece of DNA that is involved in moving from one location to another. And retrotransposons, there's an RNA intermediate involved with the gene moving from one location to another. Okay, let's look at the DNA ones, the transposons first. Here in this diagram, the light blue is the non-transposon part of the genome. In other words, regular old DNA that might be encoding your own genes there and there and there. And the dark blue is the transposon. So it's still DNA, it's still G's and A's and T's and C's, and it's still part of your chromosome. What you find if you look at the sequence within the transposon are some genes. There are some, you find a promoter and starting codons and stop codons and things like that. And the protein that is encoded by the transposon is a protein that can go back into the genome find the transposon gene that made it, and then it can either cut out a copy of that and paste it into a new location. So let me diagram it like this. Here's the nucleus. Here's the rest of the cell like that. Here's part of your chromosome. And here is the transposon gene. So the transposon gene is expressed. So it ends up making a protein product, an enzyme. And that protein comes back into the nucleus, scans along the chromosome to find its own gene, the gene that made it, and then in some cases it snips out the gene and helps it move to a new chromosome, or at least a new location somewhere in the genome, maybe a new chromosome and maybe a different location on the same chromosome, but anyway, it's going to help it move. Or in some cases, the protein goes back and copies its own gene, and then essentially takes that DNA copy of the gene and inserts it somewhere else in the genome. Like that. So, it's, I don't know, very strange thing.